Hi, my name is Carolina, and I will be talking about machine learning to filter out false positives in tumour-only NGS. So in cancer, driver mutations play a key role in developing staging systems and targeted drugs to fight this disease. However, the identification of somatic mutations is quite complex, and it's often aided by the concurrent analysis of matched germline tissue. However, in practice, normal tissue isn't always available, and so we end up with something like this, where we have a tumor sample that gets sequenced, and that will output a list of variants or mutations. Those variants can be either noise, and so that's produced by the bioinformatics pipeline or sequencing instrument itself. They could be germline variants, and so these are the variants present in all cells, and these were inherited. Or we could have the somatic variants, which are the ones that are present only in the tumor, and it's what we're interested in identifying. When we have tumor-only samples, there are strategies to eliminate germline variants to be able to separate them from the somatic ones. However, excluding noise is actually quite manual intensive and time consuming. So our aim was to develop an unsupervised machine learning model to distinguish true somatic variants in tumor-only data. So we had 62 tumor-only samples. These were splenic marginal zone lymphoma samples, and it's quite a rare cancer, so that's why we didn't really have any normal tissue. And they were processed using a targeted panel, so we only looked at 57 genes. They were sequenced using a NextSeq system, and then the sequencing data was processed using an in-house pipeline. That pipeline gave us a raw list of variants, which we filtered to enrich only for the somatic mutations. And then the variants were manually reviewed in a genomics viewer, so IGV, by 2 independent investigators, and they were labeled either true variants or false. Then we had additional information that we extracted from the BAM files, and so that included the number of amplicons covering a base, the number of mismatches across reads, and the number of reads that had some sort of soft clipping. Then we had the feature selection. So in this case, we kind of looked at all of the quality metrics that we had available and identified which ones would be informative and kind of compared it to what we would normally look at when we're manually reviewing them. Once we had done that, we ended up with 10 features that were used to run the model, and these features were scaled. Then the optimal number of clusters was obtained using the average silhouette method and the elbow method, and then they were clustered using k-means. So these here are the results of the clustering. The bioinformatics pipeline itself called over 36,000 variants, but after we filtered, only 1,367 were actually used as input into the model. And so this UMAP here shows the variants. Each point is a variant, and they are colored by the cluster, and the shape is the manual validation label, so if we thought they were true or false variants. And we can see that cluster 3 here is the one that contained the most variants labeled true. And we did a principal component analysis, and that identified that the variant allele frequency and the quality normalized by depth were the 2 features that had the greatest effect on the variance between the true and false clusters, so kind of going across. And we had a sensitivity and specificity both of 93%, so it was quite good. So if we want to apply this method to other data, we have to take into consideration the limitations and advantages. So for the most part, indels didn't do so well. In my case, I used it as a triage tool rather than a strict filter, and it was slightly biased towards variants with VAF greater than 20%. However, it did reduce the bias, and it did reduce the time that I needed to assess variants. So in my case, it was introduced into my own pipeline. So I'd just like to thank my supervisory team and the cancer genomics teams. And that's it. If you have any questions, let me know.